Hi, welcome to our virtual research innovation spotlight on cell therapy. I'm Steve Gilson with the Alliance for Southern California Innovation. We're one of the organizers of the Spotlight Series, along with our partners, BioscienceLA. Today, you will hear presentations from 4 primary investigators, each of whom will present their research for up to 10 minutes, leaving 10 minutes remaining for Q&A. We strongly encourage all the audience members to participate in this interactive session. It's not just for us and the moderators. Before we get into the research and conversation, you'll hear from Dave Whalen, the Executive Director of BioscienceLA, to say hi, and also to introduce Michael Rome from Foresite Capital, who have generously underwritten the Spotlight Series for 2023. Michael will tell us a bit more about Foresite, and then after Michael's remarks, I will ask each attendee to briefly introduce themselves before handing the program off to our esteemed moderator, Sumant Ramachandra, CEO and President of Impact Bio. But for right now, Dave Whalen, how's it going? Take it away.
Perfect. Thanks so much, Steve. Hello, everyone. Apologies for my vertical screen and weird background. You got me live from the show floor in Nashville, Tennessee, at the Vive healthcare conference right now. So fill you in more on that later. But wonderful to be here. Just really quickly, BioscienceLA is the innovation catalyst for life sciences in the LA region, and we work very closely with the Alliance for SoCal Innovation, along with academic institutions, healthcare providers, startups, founders, funders, to help grow healthcare in the region. And it's been wonderful to partner with the Alliance over the past couple of years on this program, and very excited to have a new sponsor with us, as Steve mentioned. And Foresite Capital is an organization that I've known for some time because of Michael Rome, whom I've known for even longer, going back to his days here, first time around in Los Angeles. And so, Michael, would love to have you share a little bit about what you're doing and sort of how the work that Foresite's doing aligns with our programming here.
Absolutely, thanks, Dave, and really good to be here. Nice to meet everyone. So, a couple minutes about Foresite. I'm a managing director of Foresite Capital. I'm a PhD biochemist by training, and Foresite is a healthcare venture capital and growth equity firm. We have a little under $4 billion under management across 6 funds, and I think a key feature for us is that we're a full life cycle investor, meaning we lead and participate in all stages of investment, including seed rounds and Series A's, even crossovers and IPOs. The team at Foresite invests across 3 different verticals, and each vertical has a set of dedicated investment professionals. These verticals include therapeutics and biopharma. This is a group I co-lead. We also have a life sciences and genomics effort, and our last vertical is in healthcare services, which is new for us in our 6th fund. We also have a sister incubator called Foresite Labs that launches companies on the interface of healthcare and data science. And we have 3 offices basically across the US. Our main office now is actually turned into, has migrated from San Francisco to LA. So happy to report that. We still have a San Francisco office, and we have an office on the East Coast. Probably 50% of our organization is in either the New York area, Boston, or kind of in the Philadelphia region. So talk about our team for a minute. Our team is an incredibly diverse team, and we're really proud of everyone. This includes industry experts, entrepreneurs, PhD scientists, medical doctors, leading academics, and even CFA and MBA holders. And we all pride ourselves that we really work together as a team to invest in these unique areas. On the therapeutic side, which is probably more relevant to today's discussion, we have really broad interests, including small molecule drug development, biologics, cellular therapy, which you'll hear more about today, and even a group doing computational enabled drug design kind of across our groups. Kind of the last feature that I'd like to talk about Foresite is we tend to partner with our portfolio companies. We provide a suite of services to ensure their success. This includes management team, recruitment, clinical trial execution, interaction with regulatory bodies, and even business development. Some examples of portfolio companies include 10x Genomics, which is a leading sequencing company, and also a public company under the ticker TXG. Impact Bio based in LA, and you'll hear in a minute from the CEO, Sumant. Affinivax Bio, which was acquired by GSK last year, and two notable private companies, Synthera and Alumis. Actually, both of those are based in California. So we really take pride in our commitment to supporting innovation across all sectors, which is why we're really thrilled to be a sponsor today in the research spotlight on cell therapy. And I'm personally really interested to hear what the speakers have to say, and hopefully we'll be in an interactive session. Thanks.
All right, thank you, Michael, so much, and thanks for your support of this program this year. It's been a great collaboration so far. We're looking forward to more great things to come. So now I will hand it off to Sumant actually, before I do that, so I'm going to introduce all the attendees. I forgot to do that for a moment, and then I'll hand it off to Sumant. So I'm going to go into, I'm going to skip the presenters today because Sumant will take care of that as well as he'll introduce himself. But I'll do my little Hollywood Squares invitation here and go with people who I see in order. So Walter, actually, you are the first square, and then after that will be Sonia.
Okay. Thank you, Steve. It's a pleasure to be here. My name is Walter David, and just to keep it short, I'm a director within the Research Business Development Group at City of Hope.
Great, thanks. Yeah, and I appreciate the brevity of that as well. That's what I'm looking for. So everyone followed Walter's example. Yeah, Sonia, you are up, and next will be Sissio.
Awesome, great to see everyone. My name is Sonia Satayesh, and I'm an associate at Civilization Ventures.
Fantastic. Sissio, that you're up, and then Farnaz, you'd be next.
Yes, I'm trying to connect. Can you hear me now?
Yes.
Okay, great. My name is Sissio. I'm the Global Development Lead at Amgen, Medical Director.
Fantastic, thank you. Farnaz, you're up, and after you will be Mark Mzuski. So Farnaz, you're on mute still. So still on mute? I'll come back to you. Okay, I'll come back. Okay, I'll come back to you. Okay, Mark, you are up, and after that would be Jia Chen.
Thanks, Steve. So I'm Mark Wisniewski, Senior Director of Life Science Licensing at UCLA, and glad to be here.
Great, thank you. So Jia, you are up, and then after that, we'll go with Karthika.
Hi, everyone. This is Jia Chen. I'm the Administrative Director for the Parker Institute for Cancer Immunotherapy here at UCLA.
Fantastic. Thank you for joining us. And Karthika, you are up, and then after you will be Daniela.
Hi, everyone. My name is Karthika. I work with Walter David and Daniela at City of Hope in the Research Business Development.
Fantastic.
Good morning, everyone. Happy to be here. Daniela Castanotto, Director of Strategy at City of Hope.
Fantastic. Thank you. Ryan, you are up, and then it'll be Farnaz. I think you've worked it out. Yes. Okay. So Ryan, if you want to say hi. Maybe Ryan's. Okay, well, Farnaz, you get to go now. So we'll right afterwards.
Apologies, we haven't mastered technical issues yet. I'm Farnaz Farzani. It's a pleasure meeting everyone. I am actually the founder and president of my consulting company. I serve as a strategic and senior advisor to venture capital, to startups, and to platform companies. It's a pleasure meeting everyone.
Good to see you again.
Good to
Ryan,
see you.
Ryan, are you back online yet? Maybe at the breakfast. All right. So Ryan's a good friend of the Alliance and everyone in BioscienceLA, in fact, and he's a Tech Coast Angels members also. He's been at UCLA for a bit, and now he's works at a startup. And he can't tell you that now until maybe later. I will hand it off now to Sumant, and so feel free to take it from here. Thank you for being our Steve moderator. Sumant helped actually get helped us to decide who the presenters would be from the long list of great presentations that were provided as options. But Sumant's as a credible expert in this field, and looking forward to hearing you moderate the rest of the conversation.
Great, thank you, Steve. Sumant Ramachandra. I'm the president and CEO of Impact Bio, a Los Angeles-based company. Most of our technology comes from UCLA. It's focused on what we call logic-gated CAR T cell technology. We have one asset as a therapy in the clinic right now with the UCLA trial. We're initiating our trial in the next few weeks after IND clearance. And then we have a pipeline after that. So very happy to make an impact in, no pun intended, in patients' lives through the technology that's there. If you haven't read the paper from UCLA on this lead technology, it was just published this month in Cancer Discovery. Very impressive results. And in fact, one of our co-founders is Chris, who's going to be one of our presenters. And she's worked obviously in other companies, but part of her technology is actually in this company also. And I'm grateful also for Foresite and several of the venture funds for backing us during our last Series B, and as we're going through our next fundraising in the near future. So with that, we are going to go into the main part of why all of you are here. We have selected 4 really amazing principal investigators from 4 different universities and institutions in the SoCal area, and also very grateful for Alliance for SoCal Innovation and BioscienceLA working to really make this a very robust ecosystem. Because as you'll see in any successful endeavor in a geography, you need a very strong ecosystem from venture capital and private equity, universities, people who form companies, run companies, as well as partnerships and other things. And government agencies, by the way, are actually critical to our success also. So hopefully you'll see the great science that's happening in this area, and it'll draw in even more talent and money to support that science and talent in this area. So we're going to be in the following sequence. Dr. Sol Priceman will go in first. Dr. Chris Richardson will go next. Dr. Michael Dimitriou will go third. And anchoring the presentations will be Chris Pugh-Saus from UCLA. We have a mix between more basic science and clinical applications and everything in between to really help drive this conversation today. The chat function is open, so if you don't mind, you could ask your question live. The presentations are going to be 10 minutes from the principal investigator and then 10 minutes for Q&A. We want to make this interactive. Or you can throw in the chat function, but I don't think this is a shy group, so I'm hoping it'll be a live question so we can actually hear the question and have the PI answer it. The Q&A will be after each and every PI, so it won't be at the end. It'll be an investigator presentation, the Q&A, the next investigator presentation and Q&A. So please think through the questions as you are hearing their presentation. They've all prepared great presentations. And I know it doesn't do justice to do it in 10 minutes, but unfortunately we have a 20-minute per investigator as part of this. So we'll have Dr. Sol Priceman introduce himself and then pull up his slides. They're going to be running the slides from their own computers. Thank you, Dr. Priceman. We can see it.
And you can hear me now, right?
Now we can hear you. Thank you.
Okay. I was talking to myself for a second. Nice to meet everyone. Thank you for the invitation. I'm an associate professor at City of Hope, and my lab primarily develops CAR T-cell therapies for solid tumors. These are my disclosures. And so everyone's very familiar with CAR T-cells. I'll speed through this slide, but just to say that some of my lab focuses on development of new binders that target solid tumors, and as well endowing these CAR T-cells with improved functions to target suppressive microenvironments and antigen escape, which are major challenges in CAR T-cell therapies. This is an old review on autologous CAR T-cells, which is the main focus of our lab. We have now 3 phase 1 trials in various diseases, 2 of which are licensed in prostate cancer and breast to brain metastases. The newest one in ovarian cancer, which was just funded by an R01 and is the focus of this talk. In ovarian cancer, really the treatment landscape hasn't changed much over the years. The vast majority of patients are diagnosed with advanced stage disease. Over 2/3 are diagnosed with peritoneal involvement, which is very difficult to treat and has a poor survival. We've gone after a specific target that is a pan-cancer target, but very highly expressed in ovarian cancer. It's a sialyl-TN, a truncated O-glycan called tumor-associated glycoprotein 72. And without going into detail, very selective for tumor cells and essentially absent in normal cells. If you look at the landscape of TAG72 expression, the highest expressors are pancreatic cancer and ovarian cancer, but as you can see, it's expressed amongst a variety of solid tumors, suggesting that this target could work beyond ovarian cancer, which is the disease indication we've initially gone after. And this is a summary of years of work in optimizing the CAR from the binder all the way down to the co-stimulatory domain. And we essentially landed on 3 variants, and you can see very different therapeutic efficacy in a mouse model of ovarian cancer peritoneal metastasis. And our clinical candidate on the bottom right here showing about 50% curative responses in a very aggressive tumor model. This data has been packaged in a bioRxiv paper you can find online, but has landed in an open IND now, open to enrollment as a phase 1 in treatment of platinum-resistant metastatic ovarian cancer. And we're doing a dose escalation. We've actually enrolled now our first patient. And from these studies, we've actually realized that interferon gamma is critical for CAR T cells, specifically for solid tumors. This has been long-standing work in showing that interferon gamma has effects on stromal cells in the microenvironment and can benefit cellular therapies. It's critical for solid tumors in bystander effects, and these have been published most recently. Our own data has shown that interferon gamma is critical to blocking suppressive effects of macrophages in solid tumors and overcoming resistance to CAR T cells and immune checkpoint blockade. CAR T cells modulate myeloid cells in glioblastoma, for instance, work by our partners at City of Hope. And most recently showing that interferon gamma is critical for solid tumors, not liquid tumors, through a very specific mechanism, suggesting that interferon gamma and endowing CAR T cells with improved interferon gamma could be beneficial to therapy in solid tumors. And so how do we endow CAR T cells with tumor-specific interferon gamma signaling? We've actually gone after IL-12 because of the pleiotropic effects in the tumor microenvironment and its intrinsic benefits on T cells, among other cells like NKs and antigen-presenting cells. But there's an inherent issue with IL-12. It's a highly toxic cytokine given at therapeutic doses. That's been shown clinically in numerous occasions. So groups have looked at various ways to deliver IL-12 and make it more tumor specific. We actually took a different approach. We tethered IL-12 to the surface of CAR T cells and optimized this very specifically in sequence. And what was interesting and a little shocking to us was that this membrane-bound IL-12 was induced to be expressed on the surface and was inactive in the absence of T cell activation. So we called it antigen-dependent membrane-tethered IL-12. You can see that here by stimulating these TAG72 CAR T cells with TAG72 antigen, you see increased and dose-dependent surface expression of this membrane-bound IL-12 molecule. And immediately downstream of IL-12 receptor is phosphorylated STAT4 in an activated setting. And without going through the detail of this flow cytometry, we again show that downstream IL-12 receptor signaling is antigen-dependent, requiring CAR T cell activation or through TCR to engage this membrane-bound IL-12. Intrinsic benefits of membrane-bound IL-12 was shown. We also showed that the membrane-bound IL-12 could function on neighboring cells in trans signaling, and of course, improve therapeutic response in tumor re-challenge assays in vitro, showing greater T cell expansion in an antigen-dependent manner and greater tumor control in an antigen-dependent manner over the TAG72 CAR T cell alone. We showed improved therapeutic responses and durable responses in a very aggressive ovarian cancer peritoneal model where the CAR T cell alone was insufficient. We showed increased ascites accumulation of CAR T cells that were endowed with this membrane-bound IL-12, and interestingly, increased and prolonged persistence in the blood in an inactivated state. And so there's a real benefit at City of Hope that we've taken advantage of, which is local regional delivery of CAR T cells because of the ineffective T cell trafficking through systemic delivery. We've shown this in intracerebroventricular CAR T cell delivery, which is our route of administration for our primary and secondary brain metastases studies, intraperitoneal, which is the route of administration for our TAG72 CAR T cell, which is in clinic, as I mentioned, and groups have looked at intrapleural and intralesional, intratumoral administration as well. It's unclear that regional administration can target systemic disease, which is a big issue. While ovarian cancers are peritoneal, they can also go systemic in their metastatic spread. So we asked whether membrane-bound IL-12 engineered CAR T cells could effectively target systemic metastasis. And in a dual tumor model with an IP tumor and a subcutaneous tumor to mimic systemic disease, we intraperitoneally administered these CAR T cells, and we showed that only with membrane-bound IL-12 could we durably treat both the peritoneal metastasis as well as cure the subcutaneous tumors. And to expand beyond this, we built a brain and a bone metastatic model with a HER2 CAR T cell, demonstrating that following intraventricular delivery into the brain, there was insufficient targeting of bone metastatic disease. But IL-12 engineered CAR T cells could actually durably cure the vast majority of bone metastatic disease in these models associated with an increased accumulation of peripheral blood CAR T cell persistence. And so we think this is a platform for engineering CAR T cells in general over TAG72 alone as a CAR T cell in targeting systemic disease followed by regional administration, which would, in theory, improve the safety profile of these CARs. And to look at the microenvironment effects of IL-12, we built a fully syngeneic immunocompetent model. All the prior data I showed you was in xenograft human models. And you see comparable efficacy with membrane-bound IL-12 engineered CAR T cells as you do with CAR T cells injection of soluble IL-12. And you can see on the right here a body weight decline with soluble IL-12 injections, suggesting toxicities that weren't found with membrane-bound IL-12 engineered cells. You see accumulation of liver enzymes with soluble IL-12 and not with the membrane-bound IL-12 engineered CAR T cells, and systemic interferon gamma levels with soluble IL-12 injection and not with membrane-bound IL-12, suggesting safety. We also saw microenvironment effects, increased T cell accumulation in solid tumors in the peritoneum as well as the ascites, increased CAR T cell expansion, and differential changes on myeloid populations, and specifically increases in antigen-presenting MHC class II positive dendritic cells, suggesting that there's improvements in the immune suppressive microenvironment, building on the intrinsic effects on the CAR T cells. And so thank you. This is my group, funding collaborators, etc., and happy to take questions.
So you can, again, as a reminder, use the chat function. We do have one question in the chat. Brian, you can ask it, or I can ask it for you, either way.
I'll attempt to ask it on the if in case it's too stupid. What would the membrane-bound IL-12 be expressed if there was neoantigen binding, and what's the mechanism for the expression? Is it some sort of intracellular activation, or what deems it to be activated?
Yeah, so it's driven by a constitutive promoter. So in theory, it should be constitutively expressed, but it's not. We're understanding the mechanism underlying that, but suffice it to say, this happens beyond TAG72 CAR T cells, as I showed with HER2. It happens beyond conventional CAR T cells and conventional T cells. And it is inducible by neoantigen engagement of TCR as well. So it's not just restricted to CAR T cell activation, it's restricted to T cell activation. And we also show that in trans signaling in the neighborhood with T cells and endogenous T cells, that's also a requirement.
Thank you, Saul. We have one more question. Taranaz, if you want to ask it live.
Great work. I actually worked on anti-IL-12 Dragonfly's asset for a while. Great work. Good question. Two questions mainly is that we all know that ovarian cancer is a disease within a disease within a disease, and heterogeneity obviously is going to dictate response to therapeutics. So that's one question that I have. And obviously with IL-12, the potential for CRS and myocarditis, this is one of the biggest concerns with any IL-12, kind of treatment. The third question that I didn't get a chance to put in here is that have you done any kind of humanoid models to kind of address this, what you're seeing, the data in terms of either organoids or tumoroids, spheroids, to see if what you're seeing you would see in a kind of human or human models. Sorry for all the questions.
Yeah, no, it's okay. I'll address all of them. So The heterogeneity in ovarian cancer is an issue that spans beyond ovarian cancer. It's into every solid tumor, and every tumor targeted CAR T cell has the same issue. We actually believe and have data to show that IL-12 induces antigen spread. It induces, as I showed, microenvironment changes that would improve antigen presentation and targeting of this heterogeneity by eliciting endogenous immunity. That's the anticipation beyond the CAR T cell alone. I firmly believe that any targeted single CAR will be limited by antigen heterogeneity, and it's about how to use that as an insult to engage the endogenous immune system. In terms of CRS, hasn't really been a problem in solid tumors, and particularly with regional administration of the CAR T cells. And with this membrane-bound IL-12, at least in our syngeneic mouse models, we show no evidence of toxicity. In a model where we do show toxicity with just injected IL-12, but the same therapeutic benefit. So we think CRS may not be an issue, but of course, clinical studies will tell. And in terms of humanized models, all the data I showed with the regional to systemic administration, both in TAG72 CAR T cells and HER2 CAR T cells, was in a human xenograft model. We are building out humanized mouse models to evaluate that a little more faithfully.
Thank you.
Thanks for asking that question. Well, and then there's no other question in the chat. Does anyone have any live questions?
Yes, Suman, can I just ask?
Yeah.
Sorry, I just want to ask Saul a question. What's the status of the IND right now? I think you flashed that slide up.
It's open to enrollment.
It's open to enrollment. Is that through City of Hope, or do you have other centers as well?
No, it's a single center study at City of Hope as of now.
Great. And then as far as the indications, are you mentioned ovarian cancer, but are you looking broadly?
We have preclinical studies in a number of tumor types, including pancreas, gastric, breast, and others, all showing very similar therapeutic efficacy in our preclinical models. Ovarian is our lead clinical indication. We'll get a safety assessment in that setting and then move forward into other diseases.
Great, thanks.
Thanks, Michael. And just related to that, Sol, is you mentioned local regional and the potential that other T cell repertoires may be brought in. Do you need to make the cold tumor hot by another mechanism, i.e., do you have to give the CAR T cells initially to one of the major MET lesions and then follow it with maybe systemic? How do you think about delivery to get the heterogeneity of the solid tumors that you're going to tackle?
It's a good question. We've relied on lymphodepleting chemotherapy, which we've published on, that warms the tumor sufficiently to recruit the initial load of T cells into solid tumors. We do show in our preclinical studies and anticipate that the microenvironment rewiring by membrane-bound IL-12 when it will enable endogenous immunity. We do again show that in our preclinical models. We certainly think that this is a step forward as a platform to improve the microenvironment in solid tumors.
Great. Thank you, Sol. So great questions. And I think that was all captured in the recording also for folks. I think very, very comprehensive. So thank you very much for your presentation and for the Q&A. I know that if you can stay as long as you can, that would be great. And I know you have to drop off because you had a conflict that came up. I really want to thank you for your presentation and answering the various questions that were laid out. And he put his contact information there, and obviously the business development office also at City of Hope. So please do feel free to contact them if you have further questions or are interested in the technology. So with that, I will go to the next presenter, Dr. Chris Richardson. Chris, if you don't mind introducing yourself real quick and then run through the slides.
Yeah, hi, good morning, everyone. My name is Chris Richardson. I'm an assistant professor at UC Santa Barbara. My main interest is in DNA repair, and I have some academic and professional interests in gene editing as well. So let me go ahead and see if I can get my slides. work. It says my screen sharing is paused. Huh.
Because you are permitted to do it, so it may just be a local technical issue.
Yeah, I don't know what's happening here. Okay, let me try one more time.
If you have multiple monitors, it may be an issue with that.
Yep, okay, let's try getting monitors. Okay.
Yeah, you're good.
Okay, great. We're in business. Okay, everyone see my slides and hear me?
Yep.
Okay, great. So the thing that I'd like to talk about today is XHDRTs and non-viral gene editing. So XHDRTs are an improved template molecule developed in my lab. And in many cases, we can see up to a threefold increase in non-viral gene editing efficiency. So non-viral gene editing is an effective technique to change the genome of an organism in an interesting or potentially useful way. So that's kind of indicated by this fluorescent rabbit here. And so a non-viral gene editing approach often requires targeted nucleases to specify the location of the edit, and then requires template molecules which actually contain the information that's going to get put into the genome. There are a number of different approaches you can use for gene editing. We think that non-viral gene editing has a couple of advantages. First, it is very scalable. It's easy to make arbitrarily large amounts of these reagents. It's comparatively inexpensive. And in some cases, it's less immunogenic than, for example, viral gene editing workflows. Okay, so let's back up a few steps and talk about gene editing. So I'm going to define gene editing as inserting a template-derived sequence into a targeted region of the genome. And so often this requires editing reagents, so a targeted nuclease, in this case Cas9, and then template DNA that contains the sequence that we want to introduce. So Cas9 recognizes and cuts its target site, and then the cells broker this kind of complicated interaction between the target site and the template molecule. And the outcome of this is integration of template-derived sequence into the genome by this altered sequence down at the bottom of this slide here. And so there are a number of advantages of targeted insertion. And so those include being able to put the template sequence under endogenous regulation. So if you're inserting a gene or a modified version of the gene, you can put it under its endogenous promoter. You can insert into known safe harbor loci where there's not going to be any risk of altering the target cells, and it doesn't have the same risk of insertional mutagenesis that other forms of gene therapy have, where your payload ends up in a region that disrupts regulation of some other important gene. There are, broadly speaking, two ways that you can do non-viral editing. On the right branch of this slide, there's ex vivo editing. This is editing in which you take cells out of a patient. You modify those cells in some way. You validate the edits and then reintroduce those cells back into the patients. And so this is an approach that's used for the treatment of sickle cell disease or the generation of antigen receptor expressing T cells. That we've just heard about and are often used in treating cancer. The other branch is in vivo editing, where you introduce editing reagents into a patient. They find their target and perform the edit. And so most systemically dosed editing reagents end up in the liver, and so many of the in vivo approaches end up being liver editing approaches. This can be to directly correct the lesion, to treat things like PKU or hemophilia, or there are also some ideas that you could do targeted insertion of an enzyme, which would then be secreted by the liver, and so you have this kind of edit the liver to drug the body kind of modality. So whether it's ex vivo or in vivo, these approaches are going to rely on the same components. And so the first component of this is the delivery strategy. So how are you introducing these editing reagents? This can be through approaches like electroporation, if you're working ex vivo, or lipid nanoparticles in vivo. The second component is the targeted nuclease. So how are you recognizing and cutting the specific region of the genome that you're interested in? And this can include things like Cas9, CPF1, and this kind of ever-expanding array of proprietary nucleases that companies have. The third component is the template molecule, and this is where our IP lies. So this can include things like double-stranded DNA, single-stranded DNA, adenovirus genomes, or in our case, XHDRTs. And so these components often get grouped together into platforms, right? So lipid nanoparticle delivery, Cas9 nuclease, single-stranded DNA donor. And we think this is really nice because we can kind of A/B test XHDRTs against other template strategies within these platforms, which is one of the things that we've tried to do with CAR-T editing. Okay, so XHDRTs have a number of properties. They are modified DNA. They have covalent crosslinks. This seems to be robust to multiple different types of ICL chemistries. So we can create ICLs with cisplatin, we can create ICLs with sorrelin, and both of these seem to work. Both cisplatin and sorrelin are FDA approved for use in patients. There seems like there's an optimal density of these crosslinks. That optimal density is about 10 crosslinks per kilobase, and the crosslinks need to be actually in the template molecule. They work in cis. So if you add crosslinks to the actual template, you get this nice boost in editing. If you add crosslinks to another DNA molecule that's not homologous to the genome, you don't get the same boost. And so it's not like these crosslinks are just sort of like hyperactivating DNA repair or something like that. We've tested XHDRTs in cell lines. They work great. So the delivery here was electroporation into either K562s or IPS cells. The nuclease was Cas9. This was either the HBB, the beta globin, or the lamin B1 locus. And the payload size was about 2 kb. That's the size that we were putting into the genome. And so the data on this slide, the y-axis is editing frequency, so % of cells that contain the desired edit. And the bar plots are pairs between normal templates or our XHDRT. So what we see is about a 3 to 5-fold increase in gene editing frequency. And these reactions are as similar as we can possibly make them. The amounts of the editing reagents we're using are the same, the delivery approaches are the same, and the only thing that's changing is the number of crosslinks that we have on the template molecule. And so we get a big boost in cell lines. We also get a big boost in cell yield in primary T cells. So in this case, the delivery is again electroporation into stimulated T cells. The nuclease is Cas9. The locus is Rab11, and again, it's about a 2 kb payload. So the plot here is cell yield, and what this is the number of cells containing the edit at a fixed time point. And we think this tells us two really interesting things. First of all, it says that the toxicity of these things is tolerable, right? It's not like we're getting very high efficiency, very low viability. If we compare XHDRTs to template molecules, for example, at the 500 nanogram dose, With uncross-linked template, we see about 2.5% HR, 2.5% editing, and we see about 8%. Getting myself mixed up here. We see about 250,000 edited cells with the uncross-linked template, and we see about 800,000 edited cells with the XHDRT. And this is true at multiple relevant doses of template. XHDRTs don't appear to be more toxic than other types of templates in our hands, nor do they appear to be more mutagenic. So this is data from an experiment, again, electroporation introduction of these reagents, Cas9 is the nucleus, and beta globin is the locus. In this case, we're looking at a SNP payload, so we're just altering a few nucleotides. I believe this was a wild type 2 HBB edit. So the background, the y-axis on this graph is mutagenic potential. So it's we've basically taken out all of the desired edits, and we're only looking at the cumulative frequency of mutation across the region that we edited. And so this is basically a readout for the amount of unwanted genetic change at this particular locus. Our unedited cells have this baseline mutagenic potential of 0.1%, and cells edited with uncross-linked template show this mutagenic potential of about 0.2%. And the important parts of this graph are that with increasing number of crosslinks, we do not see increases in the amount of mutation at this locus, nor do we see increases in amount of mutation when we compare cells edited with uncross-linked template versus cells edited with cross-linked template. Okay, because these are covalently modified DNA molecules, it gives us the opportunity to do some interesting things chemically. So basically, we can functionalize these crosslinks, and to date, we've done two things. So we've added biotin groups to the crosslinks. This is useful for purifying these molecules. It's also useful for recruiting things to them. And we've added fluorescent dyes, in this case Cy5, and this allows us to see sort of the intracellular localization of these reagents and track them during experiments. This plot here just shows the nuclear fluorescence about 24 hours after the introduction of these reagents. And we think this is really potentially very powerful because you can add additional functional groups onto these modifications. So from a delivery standpoint, you could add things like cell-penetrating peptides or NLSs. You could try approaches like affinity tagging your XHDRT to your nuclease, for example, or you could add groups that increase the stability or alter the chemistry in favorable ways. So for example, adding PEG polymers or something like that. Okay, so to summarize, I've told you about XHDRT molecules. This is an improved DNA template. It has higher efficiency. It has acceptable toxicity. It is non-mutagenic and chemically tractable. We've validated this in the context of CAR-T editing, and we see that it boosts edited cell yield. And we're very interested in developing this for liver editing applications, either direct correction of lesions or knock-in of the enzyme into knock-in of enzymes into safe harbor loci. And because I'm an academic, I'm required to tell you what we're doing next with this. So we're really interested in the mechanism of this. One approach that we've taken is to take biotinylated XHDRTs and basically pull them out of cells during the process of gene editing and ask what proteins are bound to the XHDRTs. And we think this is going to be a really powerful approach because potentially it allows us to generate a version 2 of these molecules where instead of having crosslinks, we can now directly recruit responsible repair proteins and reintroduce these complexes into cells. So basically get the boost of XHDRTs without having to put the covalent modification into template DNA. That's it for my talk. I'd like to thank the people that did the work. I'd like to recognize Julian. He's a lab manager and staff scientist in the lab. And I'd especially like to recognize Hannah. She was the graduate student that led this project. Hannah and her colleague Nora are currently participating in business plan competitions around the idea of and they're the name of their organization is Particle. You'll notice that I stole their logo here for many of the slides in this talk. This work was recently published earlier this year in Nature Biotech. The publication is here if you're interested, and I'd be happy to talk more about this at any time. My email is chrisrichardson@ucsb.edu. I'd also like to thank UCSB and the NIH for supporting this work, and I'm happy to take any questions.
Thank you, Chris. Great work. Right now, there's nothing on the chat, but let's just going to check. I do have one question, but I want to wait if anyone in the audience has a question for Chris on the work he just presented.
I do have a question. Thank you for sharing the wonderful work, Chris. How long of a base pair can you include in this template as a knock-in?
Yeah, I don't know. That's a great question. I mean, it's I think the longest we've ever tried is about 3 KB. We haven't found an upper limit.
And do you see any kind of efficiency dependency on the length of the insertion?
Yeah, that's another good question. You know, the way we've been testing this is just A/B testing uncross-linked versus XHDRT molecules. You do see a length dependence with editing efficiency. So it seems like the larger the construct, the lower the integration efficiency. And I think we see that as well.
Great. Thank you.
Yes.
Thanks, Shia. Any questions from anyone else? So one question for me, Chris, is there a special DNA repair pathway that's involved here? The reason, and a big reason I ask is because one of the issues with current gene editing approaches is with double-stranded DNA breaks, you get a lot of other issues, and the FDA is particularly very sensitive in this area. So if this is a enhanced approach, I could see it having even broader applications than one you laid out. Any thoughts?
Yeah, nice question. So I did put together a slide because I thought this question might come up. So we do think this is distinct from other forms of HR. So it seems to require a number of members of the Fanconi anemia pathway. So if you look at the plots on the left, you have uncross-linked versus XHDRT editing, and you can see these big decreases with knockouts of genes in the FA pathway. And this doesn't seem to influence editing with uncross-linked templates as intensely. It also seems to be uniquely dependent on the kinase ATR. So ATR is a major driver of DNA repair responses in cells. And so with uncross-linked molecules, we see basically no change in our ATR inhibitor treated condition versus our untreated condition, whereas it basically eliminates the editing we get from these XHDRTs. And so we do kind of think that it's working through maybe a parallel pathway to what the canonical HR pathways that are used in gene editing.
Okay. Thank you. Any questions from anyone in the audience? If not, I have one more. Where would it work? You've described all the places it could work.
Yeah. So you're asking specifically what tissues would it work in?
Yeah, tissues, cell types, areas that, where this particular approach would not work.
Yeah, so we kind of think about this in two different ways. The first is, delivery. What tissues can you deliver it to? And it's fairly easy to it's fairly easy to treat blood, and it's fairly easy to treat the liver. There's another consideration as well, which is whether or not these tissues are currently expressing the repair factors that are needed type of repair. So we don't fully know the dependencies at this point, but there is there may be some difficulty treating brain tissue, for example, using this approach, but we need to do more work to figure out exactly what the pathway was and the relative abundance of these different repair factors in that particular cell type.
Great, thank you, Chris. Any last opportunities to ask questions to Chris? Appreciate it. If folks have questions for Chris, he provided his email address, and he said UCSB, so I'm sure he's in the faculty directory also, if you want to drop him a note around this really fascinating and emerging area of so thank you so much, Chris, and you're welcome to stay obviously through the whole session. It would be great, and you're also welcome to ask questions of the other PIs in their presentations.
Great, thanks a lot.
Thank you. So we will now move on to Dr. Michael Dimitri, and please do introduce yourself and what you're going to talk about, so that you'll be running the slides. Perfect. Thank you.
Hi, great. I'm glad to be here and tell you about what we've been working on for the last 7 or 8 years, which we call GLITTER technology, which stands for glycan-dependent T-cell recruiter, which we think is a novel immunotherapy for cancer. So there's a number of limitations for targeted immunotherapies, either bispecific proteins or CAR T cells. So one is that many cancers, particularly solid cancers, lack safe targetable proteins. And even if you can find a target, you're going to typically have to make a different target for different cancers, and this increases cost and time of development. And third, current technology in general cannot target the most abundant and widespread cancer antigens known, typically what we call tumor-associated carbohydrate antigens. And what I refer to that as pure N-glycans, O-glycans, or glycolipids, as opposed to glycopeptides. If one goes back to their basic cell biology and looks at this electron micrograph of a cell, in this case is a lymphocyte, this has been stained for sugar, and you can see the so-called glycocalyx dominates the cell surface of all cells, and virtually every cell surface protein is glycosylated. And as you can see here in this X-ray scattering gram of an example protein, these glycans, these are N-glycans here in purple, are major structural features of each protein and readily available to be targeted. And it turns out that changes in glycosylation are a well-known hallmark of cancer and leading to high density of these tumor-associated carbohydrate antigens, or TACAs. Now, there's many different types of these tumor-associated carbohydrate antigens, which I show here. Saul talked about this one here called SILTN, but we're focused on two, one called beta-1,6 branched N-glycans, which is specific to this specific linkage here in N-glycans that are markedly increased in cancer. And then the TN antigen, which is also a truncated O-glycan. The beta-1,6 branch glycans were previously untargetable, and the TN antigen has been attempted to be targeted, but not with success as yet. And there can easily be 100 to 1,000 times more of these TACA binding sites than typical protein antigens. And so the reason why that's been difficult to target is that, for example, there's never been an antibody made that can bind beta-1,6 branched N-glycans. And typically you can get high affinity antibodies to TN antigen that are glycopeptide specific rather than the pure glycan. And so our solution to this problem was, instead of using antibodies, was to use sugar binding proteins to target these glycans. And so here's the concept. We've made both CAR T cells, but here I'm showing the bispecific proteins, where you take a single chain variable fragment from an antibody to CD3, fuse it to a sugar binding domain that's specific to a glycan target to induce killing. And this, we made two targets, one GLITTER1 for the beta-1,6 and one GLITTER2 for the TN antigen. And one of the key and critical differences between this technology and using antibodies to target cancer cells is that we have designed it to be driven by avidity binding rather than affinity binding. And by avidity binding, I mean, rather than lock and key, which is your typical protein-protein interaction, that you're using so-called Velcro-like binding. And so if you think about Velcro, what is Velcro? It's a bunch of fuzz. And then on the other end is a bunch of little sticks in an array with a little hook on it. And if you had one little hook that went into the fuzz, it would be a very weak interaction. However, if you multimerize or have many sticks and you go into the fuzz, then you get a very strong interaction. And so at a very superficial level, this fuzz Velcro looks a lot like the glycocalyx. And the idea here was to make our targeting where we had multivalency that could target these glycans in the glycocalyx. So, and this leads to two critical differences with antibodies. So target density drives activity rather than presence or absence of the target. And we think this is a way to significantly enhance safety because unlike high affinity antibodies that will bind even low density normal tissue, glitter should not. And so here is some example data for glitter-1. This is the bispecific protein. This is a killing assay, and this is with multiple myeloma. And you can see in the presence of human T cells, we get nice killing, but not in mouse T cells, confirming this is T cell dependent killing. And then we can take this exact same glitter-1 protein off the shelf, so to speak, not only kill multiple myeloma, kill AML, kill leukemia, pancreatic cancer, colon, non-small cell lung cancer, prostate, ovarian, breast, glioblastoma, and I can go on. So every single cancer type that has high density of the target is potently killed. And as you can see here, we have engineered this after many iterations so that the EC50 is in the femtomolar range or even below 100 femtomolar at its most potent. And we have similar data for glitter-2 that I'm not going to show, although it's not quite as potent as glitter-1. This is an example of in vivo data. This is an ovarian cancer model. And you can see here, when we give glitter-1, we get marked tumor regression. And then similar for glitter-2. Glitter-2 is not quite as potent as I mentioned. And this is just the quantitation of the data. So we then asked, we wanted to confirm whether this concept of density-dependent targeting using glycans does provide a major safety benefit. So what we did is immunohistochemistry or tissue cross-reactivity study to look for the highest expressing human tissues. And we found the 3 highest normal tissues were kidney, small bowel, and stomach. And importantly, we showed that these were similar or even higher in mice. And so we did a toxicity experiment in mice where we gave the mice human T cells, then treated them for 2 weeks with the glitter proteins at the same doses where we saw robust tumor killing. We saw no overt clinical toxicity. There was no change in liver function, kidney, or pancreatic function. The complete blood count and electrolytes were all normal. And when we looked at the histopathology of the highest expressing tissues, there was no treatment-dependent damage. Another way to look at this is that we looked at, was there any changes in human T cell or B cell frequency in these mice? And so the treatment didn't change their frequency. There was no evidence of T cell activation by treatment, either by looking at CD69, CD25, or PD-1. And there was no upregulation of interleukin-6 or interferon gamma. And we also repeated this tox experiment using CD34 hematopoietic stem cell humanized mice. We've now gone on to also look at taking these same designs and making CAR T cells. And so here's an example of our glitter-1 CAR T cell. And this is ovarian cancer. And then this is an example of our glitter-2 CAR T cell. And this is in a breast cancer model. And then just an in vivo experiment here of this. Both of these are still being designed to maximize activity, and we hope to then eventually select clinical candidates from both of those. So just to summarize where we are now in terms of development, a lot of this work has been funded by in Glitter-1, the Biden Cancer Moonshot program at NCI. We've also had 2 business grants from NCI, and most recently we've also got a grant from the CERM for development of CAR T cells. Glitter-1 is now undergoing IND enabling studies as at the NCI as part of a special program, the NCI Experimental Therapeutics Program, and they're going to also make our GMP product. Glitter-2, we still need to identify the optimal lead and then have additional funding for that. And then once that IND is approved, UC Irvine is going to perform a basket trial of Glitter-1 in refractory solid cancer. We're going to be agnostic to the tumor type, so it's going to be a true basket trial of solid cancers. Then we want to further develop the CAR T cells and develop INDs for those as well, and we'll need additional funding for that. So just to summarize, Glitter bispecific proteins and CAR T cells target tumor-associated carbohydrate antigens, the most abundant and widely expressed cancer targets known. We utilize high avidity, Velcro-like binding, such that target density rather than target presence or absence determines activity, and which we think provides a significant safety benefit. A single glitter can kill many diverse solid and liquid cancers without on-target off-cancer toxicity in humanized mouse models. Glitter-1 is in late-stage preclinical development with the NCI performing IND enabling studies, and then UCI to perform the phase 1 trial. And then Glitter-2, we're need further development for optimization to finalize the clinical candidate and develop an IND. And with that, I'll just put up some of the relevant people. Ray did a lot of this work. We also have a board member and angel investor, Bob Kentart, and our scientific advisory board includes the Cancer Center Director at UCI. And with that, I'll take any questions.
Thank you, Michael. Actually, there is a question in the chat. I'll start with the chat first, and then I do have one question. From Ryan. Ryan, do you want to ask the question live, or do you want me to read it out? Ryan?
Feel free to read it. Sorry, I just have some background noise.
Great. So Ryan's asked, Michael, he's asking what target is used on here. Is it PC3, SCOF3, luciferase in slide 6? He's referred to slide 6 specifically, and was wondering, is it the same target that you're looking at?
What cell line?
Well, I think that's the question, is you had multiple cell lines you studied this in. Were those all different targets, or was that the same target killing
all the same target?
And is the glyco is that pattern? So I mean, I wasn't aware that it was so widely expressed. And do you have any data on PDX models where the heterogeneity might be greater?
Yeah, so if you look at by immunohistochemistry, there's published data on heterogeneity for beta-1/6 and also for TN antigen. Roughly about 40 to 50% of high density expressing cancers are homogeneous for target expression, and then about 50% are heterogeneous. So it is a highly homogeneous target. Not only that, the density of the target increases as the cancer becomes more aggressive, and so there's data that metastatic lesions are even higher density and more homogeneous than primary. And so, and I didn't go into the data, but both of these targets are very important for the tumor biology. So the beta-1/6 branched N glycans, it drives receptor tyrosine kinase signaling tumor growth and metastasis. And so there's a major driver to express these at high levels. And if you were to downregulate it, which is always a risk for tumor escape, that would actually be a significant disadvantage to the tumor, and even potentially prolong life just by downregulating it.
Thank you, Ryan. Thanks, Michael, for addressing that. Any other questions from the audience?
Yeah, I guess I had two quick ones, Sumant. You commented on the avidity binding for these sugar binding proteins, but can you tell us what the KD is, or the affinity just for binding?
Yeah, so the KD is not really relevant, and that's because, for example, for binding to cells, we can't reach saturation. There's too many binding targets. What happens is you'll kill the cell before you even get close to saturation. It's the multivalency of the binding protein to the really high density of the glycan target, which I should point out, protein targets can't do this because they're just not nearly as high in density. You can improve avidity binding for protein-protein interactions, but not to the same degree as glycans because their density is just so much higher.
And these sugar binding proteins, they are specific for these branches. Like
Yes, highly specific. I didn't show the data, but yeah, if you knock out the gene, for example, that makes the beta-1/6, you eliminate binding.
Got it. And then on the you showed that experiment where you had humanized organs in the mouse model. But if you just were to look at kind of thinking about expression on healthy tissue, if you just did histology on human lung or pancreas or any other organs, what would the IHC score come back as? Would it be strong, or how do we think about specificity?
Yeah, so lung was negative, but the three highest were kidney, small bowel, and stomach. The kidney was really the glomerulus and the tubules. The small bowel was the microvilli of the lumen, and the stomach was the surface epithelium. And I would probably say there probably would be in the plus range. But there's probably several factors going in why those are also not targeted, because the lumen of the bowel is you're not really going to access from it's only the microvilli of the small intestine, for example. So T cells really don't have access to that, and they're not going to have access to the tubules either. But I should say, despite this expression, we see this major difference, and it's the much better way to quantitate these is by flow cytometry, which we do, and we have a pretty good idea of relative to, say, a normal healthy cell, what the density needs to be in the target, and we're trying to make that more quantitative instead of using IHC, to use immunofluorescence, because IHC often you get this abnormal enhancement of signal that can actually give you a false sense of what density is.
Got it. Yeah, and just to comment on your clinical trial, I know you said you're enrolling a basket design. Is it worth thinking about or enriching for histologies where you might find higher expression? I worry in it. Yeah.
So I have a slide on that. There's going to be two phases: a dose escalation phase where we're going to be agnostic to the expression, and then once we find the recommended phase 2 dose, we'll do a dose extension where we're going to look at high density. We're going to use immunofluorescence, positive tumors to look at initial safety. And then
thank you.
Phase 2 we may narrow based on what we see and depending on various cancer types.
Thank you, Michael, for asking the question, and also Michael for addressing it. I know we have one more speaker. I just want to make sure we give her enough time. One last question. Do you need a companion diagnostic with this for future therapeutic use? And in terms of identifying the right patient population?
Yeah, which is essentially what we're developing in parallel, which will be, I think we're planning on using an immunofluorescence of biopsy tissue. So that'll need to be.
Co-register. Got it.
Pardon?
Okay. You have to co-register the companion diagnostic with the therapeutic is probably to identify the right patients.
Right.
Thank you, Michael. Really great work. And I'll probably drop you another question on the chat or directly communicate. I'm just curious about the survival. What is the survival benefit of having these glycocalyxes in cancer? So just it's one area, but would love to chat with you offline. Thank you.
Thank you so much.
Yeah, my pleasure. So now we move on to our anchor presentation, and then we'll have a few minutes to the end. Chris, if you saw, as I mentioned, is also involved with Impact Bio and other companies, and I've gotten to know her very well. And we have calls actually with our founders every Thursday where we discuss deep science. Sometimes, even though I have an MD PhD, it goes over my head. So Chris, you're going to translate all the great work you've been doing in the recent publication, obviously for the audience and therapeutic potential. So with that, if you don't mind introducing yourself and the topic you're going to talk about.
Thank you so much for the introduction. So hello, everyone. My name is Cristina Puigsaus, and I'm an assistant professor at UCLA. I started my lab not long ago, and I've been fortunate to actually been involved in the design of several technologies that have been licensed, and I've worked in close collaboration with several biotech companies in the LA area and also San Francisco area. So today I'm gonna be presenting some results that just got published last week in Nature, and I think we can discuss the potential applications from these technologies. Let me see if I can share my screen. Do you see my screen?
Yeah, now we can see it. You can go full screen.
Yeah. Okay. So here are my disclosures. I'm a co-founder of Impact Bio, and I've licensed several technologies as well. And so my lab mostly focused, like many other people in the audience, on how to design T-cell therapies for solid tumors. At this point, it's clear that we can cure blood cancers with T-cell therapy, mostly CAR T-cell therapy, but developing therapies for the solid tumor space is quite more challenging. So, of course, my lab is worried about identifying new targets that we can use, overcoming the immune suppression microenvironment, improving the trafficking, and increasing increasing T-cell persistence and avoiding exhaustion. And so, when we think about what can we do to improve or to design the ideal therapy, actually, one thought that come to mind was like, well, the body consistently does that, so we don't really need to be creating crazy ideas. Well, you can basically just go look how we can cure people with immune checkpoint blockade just by blocking the inhibitory molecules that happen in the tumors, and then look how these T-cell responses actually eliminate completely solid metastatic lesions. So what we did in this project was go to our patients treated with anti-PD-1 therapy, take some group of patients that responded to therapy, a group of patients that did not respond to therapy, and then collect longitudinal samples from T cells from the tumor and T cells from the periphery, from the peripheral blood, and look how these T cell responses specific against the tumor, basically how they were able to complete their job and completely eliminate the solid lesions in the patients that responded, and did not do it in the patients that did not respond to therapy. So to do that, we focused in looking at the neoepitope specific T-cell responses, because they are the main target of the T-cell of the responses that induce clinical benefit in patients treated with anti-PD-1. And what we did a very close collaboration with PAC Pharma to try to isolate and characterize these responses. So to utilize the technology developed by PACT, we first started with taking biopsies from the patients. We sequence the tumor and the normal match control, and we identify the neoepitope mutations that appear in these patients. We select those ones that were actually expressed in the tumor, and then using prediction algorithms, we identify those neoepitopes that were more likely to be presented by the patient HLA. Then at PACT, they do these trimer molecules. They are basically each of the HLA molecule of the patient together with the neoepitope that you predicted. And then basically you build libraries with each of these HLA peptide molecules from all the different targets that you decided that you wanted to screen. So in this case, we screened 243 targets per patient. And then once you have these MHC molecules built, what you do is multimerize them and add a DNA barcode. So by multimerizing these molecules, what you do is increase the affinity for the T cell. And then with the DNA barcode, you allow to keep track of the reactivity of each of the T cells. So what we do is then build libraries that contain one of these capture reagents. I hope you can see my pointer. So basically, we build one of these capture reagents for each of the neoepitope mutation that we want to look for, that we want to screen for. And then we have libraries with 243 capture reagents, and we incubate them with samples from TILs and PBMCs collected longitudinally over the course of the treatment. Then we isolate single cells, and we basically put one of each single cell into a different well. And in each well, we sequence the TCR and the DNA barcode. So what we do with this approach is that we have these sequences of the full TCR alpha beta pair together with the DNA barcode that what allow us is to keep track of the reactivity. So by the end of this process, we come up with a list of TCR sequences and a neoepitope binder. Then to validate that these TCRs are actually reactive against the epitope that we selected for, we use non-viral T-cell gene editing, a similar approach than what was described earlier this morning by Chris. We use non-viral T-cell gene editing to replace the endogenous TCR of healthy donor T-cells by the sequence of the neoepitope-specific TCR that we isolated, and then we confirm the binding of these modified T-cells with the MHC peptide molecule, the capture reagents that I just explained. So with this, we come out with a list of all the neoepitope-specific T-cells that are present in these patients. And so we did this approach in 11 patients, 4 of them without response to therapy and 7 of them with response to therapy. And here I'm going to show you some of the properties of these patients. So first of all, I'm going to show the numbers of non-synonymous mutations. So as you can see, the patients with response to therapy in general have higher number of mutations, and this is nothing new. This was totally expected. And then from all of these patients, we select a similar number of mutations to screen for. And the first surprise that we had is that both in patients that responded to therapy and the patients that did not respond to therapy, we were able to isolate neoepitope-specific T cells. So our thought was that the patients that did not respond to therapy, they were not going to have these neoepitope-specific T cells, but that's not true. They do have them. They have them, and they actually target several mutations, not only one. The other interesting thing is that the number of mutations targeted is not proportional to the number of total mutations in the tumor, but it's always limited to a very small number of mutations that are very immunodominant in each patient. And what was different between the patients that responded to therapy and the patients that did not respond to therapy was the actual number of TCR clonotypes. So in the patients that responded to therapy, the immune system is able to actually induce a polyclonal response with multiple T cells with different TCR affinities targeting the same mutation. And we see that this does not happen in the patients that do not respond to therapy. And so let me go a little bit more into more detail in one of these patients, and they all look pretty similar. So here in the bottom panel, I show all the different neoepitopes that we screen for. They are ordered from left to right based on the affinity or the predicted affinity for the binding to the HLA of the patient. And then the y-axis show the level of expression by RNA stick. And then the colors that are highlighted with a different color, the bars that are highlighted with the different colors are the neoepitopes that were actually targeted. So these are the immunodominant mutations. And from each of these, here we show the different, in the top panel, we show the T cells that we isolated. So what we show here is that for this mutation, there is a mutation in IL-8 presented by HLA-A3. We were able to identify 6 different TCR clonotypes. The different clonotypes are marked with different colors. And then each of these little squares is equivalent to a T cell, while the crosses are equivalent to 10 T cells. It's just a simplification of the representation. So what this shows is that in this sample from this patient, we identified 14 cells that are with this red clonotype that target the IL-8 mutation, and 6 cells from the pink clonotype, 4 for the green clonotype, and so on. Additionally, we also identified T cell clonotypes that target the same mutation, but presented by a HLA-24. And for this, there were 2 different TCR clonotypes. And then we also identified T cells that target 2 different mutations. But as I mentioned, what we wanted to see was how these responses evolve over the course of treatment. So here are the responses on the PBMCs at day 14 after treatment. These are the PBMCs at baseline before we started PD-1 therapy in the tumor. So basically in the tumor, you already had these neoepitope-specific T cells that evolve and proliferate over the course of treatment. And what was interesting is that actually the different T cells that were there at the beginning, they are actually not in the tumor at the end. They got exhausted and they disappeared, but other clonotypes of T cells expanded, targeting the same mutations, which highlights the importance of the mutation itself, the immunodominant mutation itself, and then of this pool of circulating T cells that are found in the periphery that get into the tumor after the treatment with anti-PD-1. And these are the ones that actually probably are the ones that are able to induce a response. Very similar profiles we saw in the 7 patients with response to therapy. Here I'm showing you another one where we see already baseline in the blood and TILs clonotypes of T cells that expanded, contracted over the course of treatment. And then what we did, because we had the sequence of these TCRs, was engineer neoepitope-specific T cells using healthy donor. Basically, we took healthy donor T cells, replaced the endogenous TCR by the neoepitope-specific TCR, and tested the reactivity of the TCRs against tumor cells isolated from each of the patients. And here I'm showing you an Incocyte assay to show cytotoxicity. In the bottom panel is a co-culture with T cells targeting an irrelevant mutation. And as you can see, the tumor cells that are marked in red grow over time, while in the top panel, that is a co-culture with one of the TCRs targeting this IL-8 mutation, you see that at the beginning, you have melanoma cells in the well, and then the melanoma cells get killed. And this is how we can quantify this. You see that the melanoma cells target co-culture with an irrelevant TCR, they keep growing, while the ones that are co-cultured with the IL-8 a specific TCR, they kill. And basically, this is how this graph looks when we add all the 14 TCRs that we isolated from this patient. They are all cytotoxic, showing that they are all responsive of this anti-tumor response. Very similar results were observed with the different TCRs from the second patient that I just showed you, where most of the TCRs have a very strong killing activity, while there were a couple that had intermediate activities and 2 that were not cytotoxic. And we are also doing some functional studies to understand better the TCR biology on this. But then the obvious question is like, okay, so what happened with the patients that do not respond to therapy? So this is what happened. In these patients, we were still able to identify neoepitope-specific T cells, both in the periphery, in the blood, and also in the tumors. However, what happened is that these responses did not expand in these patients. So they were there, but PD-1 was not what was blocking them. So when you treat them with anti-PD-1 therapy, these responses do not expand. But those T cells are still there. So you can still get the TCR of these T cells, engineer them into healthy donor T cells, and test the reactivity against the tumor cell lines. And so what we saw is that even in these patients that did not respond to therapy, you still have these TCRs that you can use to engineer a higher number of T cells, and these TCRs are all still cytotoxic. So this was the basis of this. We've done this for like 11 patients. This data is was published last week in Nature, so you can look into more details for more patients. I just wanted to show a short summary for this presentation, but the main conclusions are that neoepitope-specific T cell responses to melanoma induced after anti-PD-1 target a limited number of immunodominant mutations, both in patients with and without response to therapy, but that in patients with response to therapy, these T cells are polyclonal and recurrently expanded over time. And this polyclonality has actually a lot of implications in that when you are thinking about designing T cell therapies for solid tumors. We also show that neoepitope-specific T cells can be isolated both from TILs and from PBMCs, and can be engineered into healthy donor T cells, or in for potential clinical applications, they could be engineered into PBMCs from the same patient, and that they still show reactivity against the tumor cell line, both the ones from patients with and without response. And then finally, we saw a very close correlation between the neoepitope-specific T cells in the tumor in the blood, and in the blood, highlighting that we could potentially use the blood as a minimally invasive source for monitoring and isolation of these neoepitope-specific T cell responses. And I just want to acknowledge, this is my lab. It's pretty new, but it grew pretty fast. And these are my sources of funding, which are covering a lot of the interest in our lab. We also, most of the work that we do is in developing engineered synthetic biology, and we also translate to the clinic thanks to the CERN funding that we got. So we have a lot of things going on. I just wanted to show you a brief part of it.
Thank you, Chris. Appreciate it. Fascinating work that was recently published and all the ongoing work you're doing in this field. So we do have a question actually on the chat function. Farnaz, would you like to ask it live?
Very interesting. This is such an interesting area. I mean, area of polyfunctionality and clonal expansion. Responsive therapy has just been really engaging for the past couple of years. So my question is that when I do TCR clonality, and I always do that establishing baseline with my patients as well, did you by any chance, I assume you have access to the prior treatments these patients have received. Did you see any kind of correlation between, for example, the type of treatments that they had received with the results that you're seeing? You know, because we know that some of these prior treatments, you know, they create kind of their own TCR signatures. Did you see anything like that in terms of, no?
I don't think we have enough patients to be able to make correlations. So there's only 4 patients that did not respond to therapy. So I think it's difficult to conclude why they did not respond. So we can just show that they had T cells, that they are neoepitope-specific T cells, but there is not much more to conclude other than these T cells did not expand, and there's many reasons why they would not expand. So the idea behind this is that what I would do to these patients is take these TCRs to non-viral T cell gene editing into PBMCs. That's actually the goal of Pac Pharma, and then reinfuse them back. And those T cells have the potential to actually be cytotoxic, right? I mean, as we showed. So that's what I would do. Conclusions on why the patients did not respond, I think that's too small of a cohort to actually conclude.
Yeah, and it would be also interesting if you ever had access to their pre-treatment biopsies to kind of establish a baseline that way as well. And then when you try to reinfuse them back, because you're going to have, you know, patient-to-patient diversity in terms of how they're going to have this expansion and all of that too. It would be interesting to kind of have a prior notion of how they might respond to that TCR therapy. But very interesting results. Thanks for sharing this.
Thank you so much.
Thank you for asking. Any other questions, live questions in the audience? I don't see anything else in the chat, but if not, I do have a question. Okay, so you have licensed obviously this technology to Pac Pharma, but where else outside of what Pact is doing do you see that applications of this particular technology, particularly if it can be used for predicting response to therapy, as an example?
So I think this technology, well, I think it's incredible what this technology can do, because be able to isolate T cells and know the reactivity and be able to have the TCR and put it into T cells and expand them and do therapy, I think that has a lot of potential. This is all new technology. Basically, everything was developed in the last 5 years. And so it's very new technology. I think it still needs to be improved. But from the results that we actually published in this paper, I think that what is very important and that what we should be thinking about is which are the right antigens to target when we are talking about personalized therapies, right? So the field of cancer vaccines with neoantigen peptides or now RNA oligos, they just take whatever they find the mutations, they take whatever the algorithms say, they take top 20 candidates and they do a vaccine. But what we see is actually that's not how the natural T cell responses work. The natural T cell responses target very immunodominant epitopes that are not the one that are that have higher binding affinity or higher expression. That's not how the immune system chooses which of the mutation to target. And we see that that's actually a very specific characteristic of the immune response, because in concept, like even during the course of treatment, you still keep targeting the same mutations, even though the TCRs are not the same. So being able to predict this immunodominance is going to be very important in terms of designing vaccines and designing personalized TCR T-cell therapies, right? Because instead of screening 200 mutations, you could just go and screen 10 and find the TCRs as well, right? So I think that that's probably the most important take-home message from the actual data that I just discussed, besides obviously the potential applications for direct applications for the clinical, as adoptive T-cell therapy as they are.
Thanks, Chris. Any other questions for Chris? I have one more. TCR repertoire is limited usually in all of us, right? We all have different, up to 1 million or so TCRs per individual. Maybe I'm slightly off by the number. If you have a synthetic TCR, if you have identified the TCRs for the dominant neoantigens that you're targeting, does that lead to then, when those cells get killed, epitope spreading to allow the innate T cells with their TCR repertoire to come and kill the rest of the cancers? You're targeting solid tumors. Melanoma is immunoresponsive, but there's so many tumors that are not immunoresponsive. Just a question around how you could benefit a broader group of patients with different tumors.
I think that that's a big problem, right? Like we are targeting, in our case, we look at melanoma, and you know that it's very easy to find. I mean, very easy. You know that they respond to anti-PD-1, so you know that the immune system is generating a T-cell response against those tumors. And actually, when we do this antigen spread, it's interesting that you are actually not doing antigen spread. You are still targeting the same mutations. So that shows the immunogenicity of those mutations in melanoma, just because they are able to induce a response. I think that in other solid tumors with less mutations, we are going to have a much harder time. And what I feel this is telling to me is that it's not that easy to enhance this, to induce this like antigen spread and induce an immune response against other mutations, because they are not so immunogenic. The immune repertoire does not have they don't have T cells that are actually able to target those mutations. So if those T cells are not in the repertoire, then they are not going to go to the tumor, even if you induce any kind of damage that would, attract them to the tumor. So I think that that's a problem. And I think that we need to be thinking about T cell therapies for those patients that have tumors that are not immunogenic and that they have low number of mutations. We need to be designing T cell therapies that can target all of the cells. That's my thinking. And that's why we need to very much look for antigens that are expressed, that are widely expressed in the tumor. This think of targeting antigens that are expressed in like 10% of the cells. I think that that's hard. I think we need to be looking for antigens that are widely expressed and mechanisms of, targeting, doing double targeting so we can actually approach more cells in the tumor.
Got it. Thank you. That's very helpful.
Thank you so much.
Any other questions for Chris before I hand this over to Steve? So the really great presentations. The recording will be available. Obviously, Steve will go through that. I do want to point out that the amount of investment in cell and gene therapy has gone up dramatically over the last decade. And some of that will likely taper off over time as various approaches work and others have not worked as well. But the Holy Grail is a lot of the areas that were just covered today, which is approaches to solid tumors is still evading many companies versus hematologic malignancies. Great progress is being made. So all of this work obviously will hopefully result in us making a dent in solid tumors, and then the applications can go beyond. I can tell you that there's a lot of excitement in cell therapy right now in autoimmune diseases. Okay, just based on the publication in Nature Medicine from George Schett in Germany, and people will find other applications to cell and gene therapy outside of even the ones we have spoken about today. So it's a great area, great field, and I really thank everyone for their attention, their questions, their interest. in this, and I'll pass this off to Steve to close out. Thank you, everyone.
Well, great. Thank you, Sumant, for leading such a great conversation, and thank you to all the researchers for really all the work that you do leading up to this moment in time, but also for coming and spending some time with us and giving us some insights into what you're working on. Of course, thank you to all the attendees for your energy and time. And yes, as mentioned, we will be posting this recording actually today, and I'll send out an email later today with the link to that, as well as a link to a survey. We'll fill that out, and contact information for all the presenters and their tech transfer colleagues at the institutions they work at, so you can follow with them as well. So that's really all I have for now. Thank you again also to Foresite Capital for their amazing underwriting of this series. It couldn't be possible without them. So thank you, and for Michael, if he's still on, for doing that as well.
Yeah, thank you, Michael, and thanks, everyone. Listen, Steve, one question. If they get the link, can they forward it on to others who are not able to attend, or is it only restricted to the list of people who are invited?
Yeah, no, it's actually, we posted to YouTube, but then we put that onto our website. So yeah, we hope that others we can expand the reach of this research, so people can contact who are far and wide can contact the researchers about this. So idea is to connect as many people as needed.
Great. Thank you all. Appreciate it. Take care.
Thank you, everyone.
Thank you.
Thank you, everyone.
Bye-bye.